To survive, the human lung must continuously sweep away inhaled pathogens and particulates. It accomplishes this using a mobile barrier of mucus, propelled upward and out of the lungs by a dense carpet of beating cilia. Replenishing that barrier falls to the airway secretory cells, which must operate under a strict dual mandate. They have to maintain a steady, low-level basal clearance rate for everyday breathing, while remaining capable of an explosive release of mucus the moment an inflammatory signal is detected. These histology panels of mouse airways capture the physical evidence of these two states. In a healthy baseline state on the left, ciliated cells alternate with domed secretory cells. Notice the lack of visible mucin granules. The cells synthesize and secrete mucin at a rate that matches the basal demand, preventing any intracellular pileup. Allergic inflammation shifts this balance. Mucin production ramps up as much as 200-fold, far exceeding the basal secretion rate, and resulting in the massive intracellular accumulation of dark-stained mucin granules seen in the center panel. Finally, exposing that inflamed airway to an aerosolized agonist, like ATP, triggers the emergency response. The stored granules undergo rapid exocytosis, extruding their massive payload directly into the airway lumen. Managing this dual mandate requires a single cellular infrastructure to toggle between a slow constitutive drip and a massive emergency purge without cross-contaminating the regulatory signals. To understand the molecular machinery controlling this, we first have to look at the physical parameters of the payload itself. Airway mucins, specifically Muc5AC and Muc5B, are massive polymers. A single mucin monomer can weigh up to 3 million Daltons, making them some of the largest macromolecules encoded in the mammalian genome. This comparison scales the mucin payload against standard cellular cargo. A typical neuronal synaptic vesicle measures about 50 nanometers across. An airway mucin secretory granule is an entire micrometer in diameter. Packaging a molecule this large requires extreme spatial compression. The folded, branching chains are heavily glycosylated, with sugar side chains accounting for more than 70% of the total mass. The airway secretory cell must adapt the canonical exocytosis mechanism to physically move and fuse a payload of this magnitude. Before fusion can occur, the 1 micrometer granule must physically navigate through a dense network of apical actin filaments to reach the plasma membrane. This positioning is handled by a specific trimeric protein complex. It consists of the actin-binding protein MARCS and 2 chaperone proteins, heat shock protein 70 and cysteine staying protein. Once formed on the granule membrane, this complex physically tethers the granule to myosin V motor proteins, anchoring the massive payload to the cell's cytoskeleton. This tethering is strictly regulated. When protein kinase C phosphorylates MARCS, the protein releases from the interface of the plasma membrane, translocates to the granule, and initiates the transport process. While MARCS positions the payload at the apical pole, a separate molecular engine is required to actually breach the membrane and release the mucin. Vesicular transport relies on the coiling of a 4-helix snare protein bundle. This coiling generates the mechanical force required to pull the granule and plasma membranes together until they fuse. In the airway mucin system, the complex utilizes VAMP8 stationed on the vesicle and SNAP23 on the target plasma membrane, all structured around the scaffolding protein MUNC18B. The central QA syntaxin component is currently unknown. Researchers identify the function of these proteins by observing the physiological consequences when their corresponding genes are deleted in knockout mice. Mice with a heterozygous deletion for SNAP23 exhibit a basal defect. Even without inflammatory triggers, mucin spontaneously accumulates in their airway epithelia because the constituent clearance rate is impaired. These same mice also fail to release mucin when exposed to an ATP aerosol stimulus. The stimulated pathway is broken. Because disrupting SNAP23 eliminates both basal clearance and stimulated release, it is clear that a single, universal fusion engine handles all mucin traffic, regardless of the required secretion speed. Triggering this universal engine during an emergency requires a specific signaling cascade, beginning with extracellular ATP released from ciliated cells during inflammation. This ATP binds to the P2Y2 receptor on the epithelial surface. This docks the signal to the trimeric G protein, GQ, which activates phospholipase C. Phospholipase C then generates two second messengers, diacylglycerol, or DAG, and IP3. The IP3 travels to the apical endoplasmic reticulum, triggering a localized burst of calcium directly adjacent to the mucin granules. This calcium comes strictly from internal stores rather than the extracellular space. In neurons, calcium typically enters the cytoplasm from outside the cell. Airways rely on internal stores because the calcium levels in the thin layer of airway liquid fluctuate wildly, making it an unreliable signal source. This localized calcium spike acts as the ignition spark for the emergency purge, provided a molecular sensor is in place to read the signal. The protein responsible for reading the ER calcium burst is synaptotagmin 2, or Scie-2. It is a low-affinity sensor, meaning it only triggers the fusion machinery when it detects a high-concentration calcium spike. In Scie-2 knockout mice, ATP-stimulated mucin release fails completely. Without this sensor, the emergency pathway is severed. Despite this total failure of stimulated release, Scie-2 knockout mice exhibit no spontaneous mucin accumulation. Their basal secretion rate remains normal, indicating that the constitutive clearance pathway bypasses Scie-2 entirely. This contrasts with the priming protein Muc13.2, which binds to the second messenger DAG. When Muc13.2 is deleted, mice suffer from severe failure in both stimulated secretion and basal clearance, leading to massive mucin accumulation. This phenotypic mismatch proves that basal and stimulated clearance are decoupled upstream. Stimulated release depends strictly on Scie-2, while basal clearance utilizes a different, currently unidentified high-affinity calcium sensor to operate the same fusion engine. This schematic integrates the evidence into a unified model. The ATP signal generates DAG to activate Muc13.2, and IP3 releases calcium to activate Scie-2. Both basal and stimulated pathways ultimately funnel into the same snare complex, SNAP23 and VAMP8, built on the Muc18B scaffold. The airway epithelium achieves its dual mandate by wiring two distinct sensory ignition switches to a single explosive payload. Forcing the mucin polymer through the plasma membrane is only half the process. The polymer must immediately expand to become functional mucus. Mucins are packaged dehydrated in secretory granules. Upon release, they must expand over 100-fold by absorbing water to reach the correct viscoelasticity for ciliary clearance. The cell coordinates this expansion by using the same ATP signal that triggers the snare complex to also signal CFTR channels to pump chloride, drawing the necessary water into the lumen via osmosis. While the core mechanism is clear, the specific QA syntaxin and the high-affinity basal calcium sensor remain undiscovered. Airway mucin exocytosis depends on the precise synchronization of the massive macrocargo, decoupled genetic sensors, and instantaneous fluid dynamics. Maintaining this system is the baseline requirement for every healthy breath.